It's actually, let me go get it, it'll be better.
Okay, we have time.
Hello, hello.
Good morning. First of all, I want to have a big shout out to the students who helped me set up today. I know these are the students who always help me, and thank you so much. It is my great pleasure to introduce our first scientist for the speaker series, Dr. Carol Marchetta.
Thank you. Thanks, Sophia, for inviting me. Thank you all for coming that early, kind of early. And thanks for all the help with setting up. Oops. Just gonna walk there.
You're fine to go anywhere, just
So I thought I'd start by doing somewhat informal. Oh, first I have an accent, as you can tell. I'm not from here. I'm from Brazil. So I wanted to start maybe telling a little bit about my story, and have an idea of how much you guys know about science and stem cells, and then I'm going to tell you what is it that I do in the lab. So I came to the US in 2002 because I was interested in what the brain cells were doing. And I end up, at that time, it wasn't very easy to, for a person from a different country to just come here and find a lab and start working in the lab. So I started doing a volunteer work. So I worked as a volunteer for almost 2 years, and we lived in a house with many people. And then I defended my PhD back in Brazil. And then I came here as a postdoc. So you do a postdoctoral job after you do your PhD. And this was 2005. And then since then, after 5 years of a postdoctoral job, you do, and this is all in the research laboratory. After that, you change your status because you're not a temporary person anymore. You are a staff member. And then I became a staff scientist at the Salk Institute in La Jolla. Some of you might know. The Salk is, Jana Salk is the one that helped develop the Salk vaccine for polio. If you guys want to visit the place or want to take a look online, it's a very cool, beautifully designed building by famous architect too, Louis Kahn, which also has a very interesting story himself. So that aside, I am at the Salk studying brain cells in a dish, and one of the main questions that we had was, how can you, why study brain cells in a dish? Any of you have or know of anyone in your friends or family that have mental illness? Yes, or disease or disorder or change. This is actually something that it's very pronounced in not only the US, and you can take a look at the numbers. Autism nowadays, so those are National Institutes of Health numbers. 1 every 68 kids. Schizophrenia, 1% of the US population. Mood disorders, if you include major depression, bipolar, 9.5% of the US population. And if you group mental illness with anxiety, substance abuse, that grows into 18%. And the very important part of that is, for instance, for autism, there are no effective drugs. For schizophrenia, most of the drugs are refractory to at least 1/3 of the patients. And for mood disorders, the antidepressants only work for 54% of the patients. So that is an issue for a number of reasons. One of them is because these people could be functioning much better, and they're not, because they're not being supported in the ways they could. But mostly because you have an amount of population that is suffering, and this is not even counting the homeless in the streets, right? That a lot of homeless have mental illness, and I'm not sure if you guys are familiarized with the homeless situation in East Village, for instance. It's just growing a lot, and we can help this population. So how do you study? Okay, so why the drugs are not working? Only 90% of the candidates, of the drug candidates in the 90% of the drug candidates that a pharma company puts in the market fails. And one of the reasons why, especially for mental illness, one of the reasons why is because the biological models that are available have very low predictivity. So what that means is that if I have a model, if I have a drug that is really good on mice, for instance, on rodent, what we call rodent models, the chances that that drug is going to work in humans is very low. And that's a reason for that. And the reason is because the cells in the brains of the humans are very different from the cells in the brains of the mice, for instance. So how would you, yes?
Aren't the neurons of both all mammals more or less similar in nature because of how they work?
They do work in similar ways, but they respond differently sometimes to stimuli in different ways. And they also, the circuits that they form are different. If you look at the size of a human brain, and if you compare to the size of a rodent brain, the rodent brain is a smooth cortex, so there's no folding. And when you look at a human brain, everybody kind of knows how a human brain looks like. Lots of folds. So that means we have a ginormous cortex that does all kinds of processing, that does require some specialized cells that don't necessarily show in all types of brains. So how would you study cells, brain cells in the laboratory setting? Anyone has any ideas? Yes?
You would have to get stem cells and then you convince them to differentiate into neurons, most likely. That's how you would seem to get it. Or you could try to get neurons from affected patients, but that would require to wait until they die or some other method.
Yes, so one way you can do is you can coax stem cells. There are cells that are undifferentiated, and you can push them into a specific thing. As you what's your name? Alan? Alan. If as Alan mentioned, one way to get that done is to coax a stem cell into change into a brain cell, and then you use the brain cell, which can be a neuron, but can also be other types of brain cells. Our brain has mainly 3 types of cells, but other cell lines. So you have brain cell neurons, glial cells, which are the support. So glia comes from glue in Greek, and it can support the third kind is called the oligodendrocytes. So the oligodendrocytes, like for those of you who like engineering and wires, the best way to have one wire connecting to the other is to ensheath that wire. So the oligodendrocytes, they provide this isolation in the processes of the neurons, allowing for the signal to be transduced faster. Everybody is okay with the concept that neurons talk to each other by electrical impulses. So one way to make the electrical impulses function is to isolate that neurite that communicates to the other cells. Let's get some water here. So the oligodendrocytes, which is the third type of neuronal cell, does that, of brain cell, does that ensheathing. Okay, so as you mentioned before, a great way to study brain cells in a dish would be to make a stem cell into a brain cell. That would be okay, except that most patients with mental illness you can't really take the cells from their brain to study them, as you've mentioned too. So you can crack people's skulls and look at their brains. You can look at brain imaging, so there are some technologies. And you can infer function by looking at how, so brain imaging will tell you how the areas are active or not active. But the resolution, as you can imagine, is very small. So you cannot look at a single cell or not even at a group of cells in when you use brain imaging. So a very smart group of people in Japan, led by a researcher called Shinya Yamanaka, had an idea. So as I mentioned before, the existing models that we have are limited, right? We can't look, we can also, postmortem tissues don't represent the live cells. MRI, the resolution is small. You can look at blood cells, but then as I was telling before, blood is not brain, and you can get some information, but you miss function, and you miss live function. As I mentioned before, the rodent models are limited. So what this researcher did, and he got the Nobel Prize in 2012 for his discovery, this is Shinya Yamanaka, he found amount of chemicals or factors, limited amount of factors, that when you apply this factor to a skin cell, for instance, which is readily available, or a blood cell, for instance, you can coax this cell into become stem cells. So these cells are now called induced pluripotent stem cells because they have been induced to become pluripotent or stem, yes?
Sorry, just curious, like, how accurate are induced stem cells compared to actual stem cells, like unmodified and all that?
They are remarkably similar. That's not to say that there are not differences, but they're really close, yes. So what are the advantages of this technology now? Now I can have brain cells potentially, because now I have stem cells from a patient. But potentially now I can have brain cells or neurons, glia, oligodendrocytes, and astrocytes from patients with all kinds of disorders that I wasn't able to work with them anymore or study their brain cells. So now I can. Yes?
That's a problem though. Even if you could get a genome from a person, there is so much information in someone's genome that even now with our technology, it's hard to tell
So now we have very powerful technology that can analyze whole genome sequencing from an individual in very short time. And that helps. How to know which mutations are important? The more we sequence, the more we know what is average, what is different, what is de novo mutation, which is a mutation that happens only when you are only on your DNA, but not on the DNA of your parents. So those things we start now that the technology of sequencing, the DNA sequencing, is so advancing so fast. You can now have an idea of which mutations are the important ones. Having said that, for a lot of disorders such as autism, the percentage of mutations that are the de novo mutations is the most common factor of causing the disorders. It's one of the most common factors of causing the disorders. And the de novo mutations don't always happen in the same genes. So you do have to do a lot of sequencing to find out to narrow down the important ones. Yes?
Could we just hold questions till the end of your discussion?
Yes.
Because I'm afraid we're short on teaching. And I'm afraid that we're going to run out of time.
Okay.
Is that all right? So everybody okay with that? We'll just hold questions? All right, thank you.
Okay.
Great.
Okay, so two very important things here that I want you to take home is with this technology, we can now study potentially any cell from your body, not from the body of a person or an embryo from which we derived embryonic stem cells. Now, stem cells that we derived from a line, we can study your cells or the cells of potentially any person. So that became a very important tool for potential personalized medicine. Because your mutation might be different from the mutation of a person by your side. So I'm going to tell you an example of what we have done in the lab to study, for instance, autism. We do have what we call models in the lab for different disorders. And I'm just going to tell you a little bit of how we can use this technology to understand autism better in the dish. So the title of this talk is Using Stem Cells to Research Autism Spectrum Disorder. So I'm going to break that down. What is autism spectrum disorder? It's a neurodevelopmental condition. Impaired children with autism can have a triad of syndromes, symptoms: impaired social interaction, impaired language and communication, repetitive and restricted behavior. Over 1% of children, 1 every 68, which is around 1.5 million Americans, are living with autism and growing. A cost per child affected with ASD over lifetime is between 3 to 5 million. Autism is like an important condition that is mostly genetic, and there is absolutely no therapy and no cure. And I think cure to autism can be questionable.
I was about to say about the therapy.
Mm-hmm.
Technically, there is therapy, but it's mostly
Behavioral.
yeah, basically, mostly there's pretty much a small portion of people with autism that had early intervention, but most are usually either not diagnosed or diagnosed very late, or at least late teens. But yeah, there is. Sorry, I interrupted. But
That's okay.
Technically, is therapy.
Yes, there are interventions, and behavioral interventions do work, but there are a lot of symptoms that could potentially be treated with different types of therapies that are not drug therapies that are not explored. We will. I think it's something to discuss, right? And but I think a big issue with a lot of the mental disorders is that there is not enough biological understanding of what is going on with those brain cells. And when you don't have enough biological understanding, you can't even not only design therapies, but you don't even understand what is the cause of the disorder itself. What's the biology behind the disorder? So why is the neuron behaving one way or another? So as I mentioned before, you can't crack skulls of people and look at brain cells. So what we and other researchers in the field are doing is we're using stem cells to research autism spectrum disorder. And anyone knows what a stem cell is? Definition? Yes? Anyone? Okay. You guys. Okay, you can give one, you give another. There's two definitions.
Well, basically, in layman's terms, basically it's a building block cell or a Lego cell. Basically, it's a cell that will either be used as like a building block for either a blood cell, liver cell, nerve cell. They could be anything you want to be with certain chemical reactions and all that. But they could be used.
Okay, so it can differentiate into different, we call differentiate. It can change into different types of cell bodies. That's definition number one. What's definition number two? Self-renewal. So one stem cell become another stem cell. So there's two main properties of a stem cell. It becomes a stem cell. It divides and becomes a stem cell. If a stem cell only becomes differentiated cell, you extinguish the pool. And then you don't have a stem cell anymore, you have a differentiated stem cell. So a stem cell has two main properties, self-renewal and differentiation. Okay, and we all know that from when we were born. When after fecundation, we become a big ball of stem cells, and then those stem cells start differentiating. And that's why we have our body that is composed of many differentiated cells. I have eye cells, I have brain cells, I have all different cells. Anyone knows what happens when a differentiated cell loses its capability of being differentiated and wants to become a stem cell again? Cancer? Cancer is one way of the cell, when the cell loses the program and realizes, loses the ability of keep the same fate. Abnormal growth, it doesn't understand anymore that it should be an eye cell or a specified type of line. And what that researcher did is he used that capability of the cell to not be a defined cell. And why is this important? This is important because what Shinya Yamanaka, the Japanese researcher, discovered is if a cell line can differentiate, a cell line can also de-differentiate. We knew that in the body, but we didn't really understood all the principles in the dish. And why is this important? Because when you de-differentiate the cell, so we have all our DNA is the same. Pretty much everywhere in our body. That's there's some questions and discussions of specifics, but for general purpose, the information to make us is available everywhere. And it's just a matter of which part of the book you read. So what are the instructions? The instructions is to become an eye cell. So you open up the chromatin, the DNA part that reads brain cell. Do you want to become an eye cell? Do you want to become a belly cell or stomach cell? You open a different type of information in the book. But all the information is there. This will become very important when we are talking about stem cell therapy. And we're not going to talk about stem cell therapy today, but keep in mind, especially in the light of the paper you read with Sophia, where researchers are making chimeras or making organs in different, human organs in different animals, and or transplanting human cells, for instance, in patients that have spinal cord injury, The importance of this type of clock is because those cells have flexible fate. What that means is there is a risk that if I transplant organ on you that have came from a pig, or that it's a human. Everybody remembers what the paper that Sophia gave? So it's basically growing an organ inside the pig using human stem cells, and then using that organ to transplanting patients that have, for instance, diabetes. So grow a pancreas in the pig, transplant the pancreas into a human that has diabetes. This is great technology. Caveat, though, is because those cells came from stem cells, there is always a possibility that that cell, so you need to put a lot of controlling factors that would not allow for that cell to become a stem cell again, because it was once a stem cell. And the reason why is because stem cells can become cancer, because they divide, they are undifferentiated and they divide nonstop. So when you look at this technology, always think about, especially stem cell therapies, always ask the question, how do we know that this cell that is being transplanted into a person doesn't have the capability of becoming a stem cell again, and the stem cell can be a cancer cell? Okay, back to autism. Just because there's a lot of news in the literature, so you have to keep asking yourselves and the scientists and the doctors those questions. A lot of people are going out of the US to do stem cell therapy, and we don't necessarily know what is it that they are injecting on these people, and that could be dangerous. So we use stem cells to, again, ability to self-renew and differentiate. That's kind of how they look. They are like very round and flexible. We use them to research or to model autism spectrum disorder. And what is a model? Anyone? Hmm? Oh, sorry? A model is? Yes, a guess of how it might work. It's a way to go. Yeah.
I'll say it's a representation of
The reality.
how to practice X, because it could be anything.
Yeah, that's a good try too. So models it's a simplified representation of the reality. So this is a model. That's Giselle. That's a model. And that's a model. That's a maquette of an architect. So those are all models. And we do the same in the lab. We use a simplified representation of the reality, which is stem cells, to model disorders. So again, I told you about the changing the fate of stem cells into differentiated cells. Let me just look at the time. 8:30, okay. So as I was telling before, what does stem cell technology do is it helps you understand, when I was saying how to understand disease biology. And for us basic scientists, that's really important, because as I was telling before, you see a patient that has bipolar disorder, for instance, you don't really know exactly what happened to the brain cells of this patient before it acquires the disorder. There was a tipping point that became apparent, but there is changes that happen early in time that you don't necessarily see, because the organism may not necessarily show in terms of official deficit. But there are impairments that happen on, for instance, connectivity between neurons that are early to the official diagnosis. And for bipolar disorder, for instance, or schizophrenia, the diagnosis can take a long time. Same for autism. So if we understand what happened earlier, we may not only understand better why the disease manifests, but also we might get some insights of what are good biomarkers for the disease before the disease happens. So let's say there is a specific change that happens in your brain that you start producing a chemical that you didn't produce anymore. You might not necessarily be manifesting the symptoms of the disorder, but you might have that evidence of that chemical in your blood or in your spinal, cerebrospinal fluid. And maybe if you can, if doctors can pick that up earlier, they could approach you in a different way for even behavioral therapies, for instance, or real therapies. So that's what we call a biomarker. So I like to use this illustration to tell people what we are trying to understand here, study here in basic biology. So let's say you're walking around campus and you see a car stuck in the tree. How do you know what happened before? What were the events that preceded the car to be stuck in the tree? So that's what we're trying to do with stem cell biology. We're trying to go back in time and understand what were the very early events before the car got stuck in the tree or before the disease develops. Here it could be a joke from kids in the neighborhood that decided to put the car there, or it could be a typhoon or some weather event, but you don't know. You just see the car in the tree. And a lot of times that's what happens with mental illness and different conditions. We don't really know how you got there. And that's the part of the biology that interests me and other basic biologists that study neurostem cell disorders. So how? So you can have a better understanding, and you can also prevent it from happening if you understand what are the early events preceding that. So our model is we compare stem cells or induced pluripotent stem cells, the ones that are differentiated from patients, and there are we get them from skin biopsies, and we change the skin biopsies into induced pluripotent stem cells. And once we have those stem cells, or those induced pluripotent stem cells, because they have been induced to be stem cells, that's why we call them that, we differentiate them into brain cells. And here I'm showing the three types that I mentioned before: oligodendrocytes, the in-sheathing cells, astrocytes, and the supporting cells, and neurons, the cells that actually do the electrical activity, communication. And then what we do is we compare patient cells with what we call neurotypical or control cells, cells that comes from individuals that do not have history of any type of disorder. So we started with Rett syndrome, which is a disease that is under the umbrella of autism spectrum disorder. And first thing we saw is when we compared the neurotypicals with the Rett syndrome, is that the neurotypicals are bigger than the Rett syndrome. So the Rett brain cells are different. They are smaller. So those are the types of things that we are looking in the culture. So we put those cells in the dish, we look in the microscope, and first thing we look, size. Size is different. And when you look at the patients have smaller brains. So it's not actually the number of neurons that is different, but the size. Other thing we look is at the processes of the neurons. Neurons communicate with each other via extending processes and touching other neurons using, getting very close to other neurons using those protrusions that we call synapses or puncta. So the synapse doesn't show very well. The synapse happens when two protrusions come together. and they exchange neurochemicals. So that's one of the sites for synapses, and that's how they communicate. So the other thing we notice is they had autism spectrum, specifically Rett syndrome, had a smaller number of protrusions. Smaller size, smaller number of protrusions. And also less communication with other cells. And we can tell that by giving a chemical that will be incorporated into the sites of the communications. So we hypothesized that, so that's just a summary. So smaller cells, less points, and that's the actual image of a neurite or a processes of a neuron from a Rett syndrome patient in the dish, showing the points, showing the points of connections. That's how we visualize them in culture. So then we hypothesized that they are probably communicating less with each other. And how do we do that? Let's see if that video plays. So we give a chemical to the cells that whenever they communicate to each other, they express a light, a fluorescent light. So when we look at neurotypical cells, and you see like the spots shining, so that means a neuron is communicating to each other. And when we look at autism cells, the amount of activity in the dish was decreased. So you see, we measure activity by the percentage of cells that are blinking. So I'll play it again. Can you see? Can you all see? So the blinking cells. And when you play, the percentage of blinking cells is decreased, meaning that the cells are there, but they're not communicating as efficiently. So those are the types of testings that we can do in culture and start understanding what is happening to that neuron in culture that is not communicating efficiently to the other neuron. More interestingly, another thing we can do is to rescue the effects. So one chemical, yes? One chemical that, which is actually a growth factor that is already being used for not only Rett syndrome, but also idiopathic autism, as well as other genetic forms of autism, is called insulin growth factor 1, and insulin-like growth factor 1, and we call it short for IGF-1, and it's already in clinical trials for patients, and we wanted to see if that drug was doing was changing anything in our lines, or potentially rescue the effects. And when we do that, we see that adding IGF-1 can bring the cells to be more similar to the neurotypical cells. So IGF-1 can rescue the effects of the defective connectivity in vitro. Yes? There are no new cells, exactly. So basically what IGF, and that's our follow-up is understanding how IGF-1 is improving, how IGF-1 is improving the function of the neurons. There are some theories where IGF-1 potentially boosts a few cascades that are defective in autism, and those cascades improve synaptic function. It is a very general drug or growth factor. It's not acts in, by very general drug, I mean it acts in many different pathways. It's almost like giving more than what is necessary, but it is one way to tease out what could be the factors, the pathways downstream of autism changes in the DNA. So what could how could a neurotrophic factor improve those connections? And we can have an idea by looking at the, for instance, expression profiles of what is it that the cells were producing before. Example, the autistic cells, the cells from autistic patients, what is it that they were producing before we give IGF-1, and what is it that they are making after we give IGF-1? And by comparing before and after with neurons from neurotypical patients, we can start teasing, neurotypical individuals, we can start teasing out what is it that now they are making that is more similar to individuals that don't, neurons from individuals that don't have autism. So we're starting to make those correlations and figure out what are the important pathways that are being hit by IGF-1 and narrow down into more specific drugs. Yes? Okay. I am almost done. So what that study told us, and I think it was a very, it's an important point, is at least at that stage, the cells from ASD individuals, the defects that they were showing in vitro were not permanent, which means there is some plasticity in the system that we could tap on, which is really important. We all somehow understand plasticity in our brain in terms of, let's say, I start to do a new exercise, we can learn new things, so there is a plasticity built there in our brains. That plasticity decreases as we age. That's why we go to school when you're young, and that's why it's easier for kids to learn language early on than later on. So plasticity is something that is way stronger earlier than later. So that's why it's important to maybe improve the diagnosis. Exactly. So earlier interventions are probably more effective. And there are some, and also there are indications, at least for autism spectrum disorder, that the earlier you diagnose it, the earlier you start with behavioral therapy, the better the results. Now you can always argue that detecting autism that early is not necessarily a trivial exercise, but there are a lot of, at least two groups, one in San Diego, looking for finding making questionnaires for parents to detect in toddlers what are the percentage of toddlers that can have autism or not, based on sibling studies. So there's interesting data out there. But that's the interesting part, I think, here is that understanding plasticity at the disease level, I think it's an interesting way to go about it. Not only in the brains of neurotypicals when you learn stuff, but also when you have and when you are in a disease state, there's also a great amount of plasticity still built up there. And one way to tap on it is understanding how big the plasticity is and how much can you intervene. More understanding could lead to better therapies potentially. So again, we can use the exact same model, and we are using the exact same models in our lab to look at other neurodevelopmental and neuropsychiatry disorders, such as the more idiopathic autism types, the more common ones, schizophrenia, bipolar, Williams syndrome, and we're also looking at major depression. Williams syndrome is a rare disorder where children have a duplication of a piece of a chromosome, and so it's genetic and it's very defined. And what happens with those children is they get extremely social. So a lot of people, some researchers like to contrast Williams syndrome with autism, especially because the same exact part of the DNA is deleted in some specific subtypes of autism. So it is interesting how addition can cause hypersociability and deletion can cause autism, which is known to be a little bit less social than average. This is not the whole story, but these children are extremely social. So and they have a very good facial recognition and very good eye contact, which is also something that you don't see in autism very common. They have impaired, their IQs are lower, and even though they are very drawn to social interactions, they can't maintain. And it's a source of a lot of frustration for them.
From what I've heard about like autism, or more specifically Asperger's, basically even though they have like a low socialness, there is potential that they could have like higher IQs supposedly.
So autism is a spectrum. Yeah, so autism is a spectrum, as the name says. And when you talk to clinicians, they say when you see a kid with autism, or a person with autism, you saw a person with autism. So it's typically very non-generalizable disorder.
Sorry, I asked because I've never heard of Williams syndrome.
You should look at it's very rare and yet very interesting disorder. And one of the Williams syndrome specialists is also a Salk resident too, and she studied language in Williams syndrome patients, and she's part of the Williams syndrome National Association, and they made a few videos from those children, and what they say is, and you see the kids saying, if you have to have a syndrome, you should have Williams syndrome because it's so cool. And it is the kids are the cutest things. They are a little bit like fairy, like very loving. Yes.
So you said that we don't ever generate new neurons. Is that true? I thought we did get new neurons over time, like very slowly, but I thought we did new neurons.
So one of the paradigms, and maybe how late are we? 10 minutes. Okay. Yes, this is a very good point. The lab I joined at the Salk Institute from Fred Gage is one of the labs that propose and reinforce the idea that the cells in your brain are not static. Again, as I said, there is plasticity happening, and the dogma used to be that once you are born with your brain cells, you don't have any more brain cells. You're done. Now it turns out that that is kind of true for for most areas in your brain, except a very important area in your brain called the hippocampus, where you make new memories. So your hippocampus is the only place in your brain where new cells are still born. Again, the earlier in development, or the earlier in life, the more cells are born. There is a decrease of newborn neurons as you age, but you can boost that up by, for instance, exercise, socialization, and there are different studies now showing that you can boost your we call neurogenesis, the genesis of new neurons, by maybe taking flavonoids, or so there are some new data coming out. But yeah, there are newborn neurons. They do contribute to your new memories, because that's what the hippocampus do. Yes?
Where are you specifically looking for cells? Are you just trying to get people with different troubles or using glial cells? Or
Where, sorry?
where are you typically getting cells to get what are you calling stem cells?
Skin cells. So we usually have collaborations with clinicians, and the clinicians have specific cohorts that they select that could be potentially interesting. And because you're talking about diseases where the variability is very big, we try to get clinicians to select cohorts of patients that have similar changes. So example, patients that either respond to the same type of medicine in the case of bipolar disorder. And so then we know the cohort is a little bit more tight, and we're going to see less variation in culture. And then they give us the cells. They are all de-anonymized. We have no idea who the person is, which is an important ethical statement. Yes?
My 9-year-old sister, Emery, has Asperger's syndrome
Mm-hmm.
and is highly socially impaired, but extremely intelligent when
Mhm.
like math and science.
Yeah.
What's typical for a brain cell for her brain to look like when it's like that?
I don't think we have very good answers for that. There, especially because Asperger's is rare, especially for girls. Traditionally, it's 9-1, and it's quite rare. And there's not that many good statistics when the N is not so high. And in fact, it's something that we were talking about in the lab too. We should have get a cohort of patients, individuals that have Asperger's, because would we expect to see the same changes, or would we maybe expect to see even higher connectivity, right?
I can partially answer that question. From what I've heard, clinical tests, and no, these were like kids and all that, but from when most people think of Asperger's, when they do an activity, they usually think about low activity part in like one part of the brain. It's actually the opposite. There's like tons of activity in various parts of the brain, so there's technically overactivity, or at least that's from what I've heard.
That's one of the theories, yes. And I always find it really interesting when people say, I don't use enough of my brain capacity. I want to use 100% of the capacity. If you turn, and we have inhibitory cells in our brains, and they're very important to keep information in check, because you want to be able to focus. If you have parts of your brain that fire, if all of your brain is firing out of control, you can have seizure if they all fire at the same time, but you can also get very, very confused. And because you have a lot of inhibition, when you need some inhibition to perform your tasks. Because while you're focusing on, for instance, your test, you need the sensation of being sitting on the chair to not be overly stimulating. You need all the lights outside to not be stimulated. You need all these sounds that we are hearing, or this shh, not to be. So we work at our best functioning level by inhibiting a lot of things. So that's why it's so fine. That's why we are so fine-tuned when you're talking about brain activity and brain function. It's not that straightforward. Just enhance the brain function, or just decrease the brain function. There is a balance that you need to maintain. There's a homeostatic place you need to reach in order to function, and all of us have a different one. So yes, it's okay.
Because you
That's,
about the brain.
hmm
Sorry, so sorry, sorry.
That's okay. That's okay. Yes?
So I did a little bit of research on like IGF-1 and like I guess to consider compounds that can increase like neuroplasticity, like lorazepam, aniracetam, huperzine, all that other stuff. But I was wondering what the difference between IGF-1 and like growth hormone is. I know there's more than just IGF-1. Is there like IGF-2? Like that. There's different like growth factors. But I was wondering the difference between IGF-1 and
Different receptors, to give a short answer, meaning that different cells will respond differently because the drugs act upon a reception in the cell. So different cells have different receptors. So different drugs will act different in different cells. And I think with that, I am done for today. Thank you so much for coming.
Thank you. Thank you so much, Dr. Marchetta. You have really helped us strengthen our Internship Speaker Series, helping us augment our curriculum across the whole entire school and outside the walls of academia at a very basic level. So thank you so much. Let's join in saying thank you to her.
Oh, no,